All right, so here we've just talked about tensile strength and cavitation versus embolism and mechanisms that limit cavitation, being root pressure, and also certain plants adapted to freezing and drought, perhaps having bordered pits as a way of bypassing a blockage or cavitation. Okay, so we're looking here now again at evapotranspiration, the movement of water from soil to roots to leaves, and just talking about transpiration in general. Transpiration accounts for a significant amount of water loss from the plant. So about 97% of the water that is taken up by soil is transpired either through leaves, which is most of what gets transpired through stomata, and about 7% through other openings in the plant, the stem openings called lenticels, and then also through the cuticle itself, which is a waxy layer on the surface of the leaves that's actually geared to prevent excessive water loss. The remaining 3% of the water that's taken up from the soil is used for, we'll say it this way, used for plant growth, development, and metabolism. So only 3% is actually retained for physiological processing. So remember, the rest of this is to the other functions associated with transpiration include to circulate water, nutrients, hormones, sugars to transport around the plant. It's a circulatory system for the plant. Also to provide structure for the plant. So that sort of structural support so that plants can have those outstretched leaves for photosynthesis. So to maintain turgor. Okay. Now we talked before about how we want to make sure we can trace a water molecule through all these various structures till it reaches that apoplast of the leaf mesophyll cell, which you can see surrounding the individual water surrounding individual cellulose microfibrils in the cell wall. And what these diagrams also help demonstrate is, or remind us, that evapotranspiration has two main driving forces or steps. One is evaporation from the leaf air surface there, interface, and then once water becomes vapor, then it has to diffuse out of the area, so diffusion of the water vapor. So that's what we're going to concentrate on here in this next, oops, hold on, let's go back for a second. In the next slide. This diagram over here, just making sure that we highlight that we're looking at guard cells. This is an open, since we're talking about diffusion of water vapor out of the leaf into the atmosphere, this is an open stoma, and whereas this is a closed stoma. And one thing that we want to emphasize the importance of is that turgor pressure is critical here for opening versus closing stomata. So an open stoma has higher turgor here, higher turgor pressure, which creates that opening there because the microfibrils are actually kind of radially arranged like this. So as the turgor pressure increases, this side of the guard cell stays pretty tight, whereas this part expands outward. And then over here with the closed stomata, we have lower turgor pressure. Which means it's a little bit more deflated, and so these sides of the guard cells tend to come together more tightly. Okay. All right, so we're going to move into a discussion of those two steps, starting with the question of what drives evaporation. which reminds me of one of the functions that we didn't list back there was actually temperature regulation. So what drives evaporation? It has something to do with temperature. All right. Now, what specifically drives evaporation, and we're looking at the air-liquid interface here. So we have this interface here where we've got liquid water, and we've got air up here with lots of water molecules in the vapor state. All right, so the idea here is that water molecules evaporate to vapor, and vapor also condenses back to liquid water. So we have this sort of movement back and forth between evaporation and this one here, which is condensation. Okay, so the vapor that accumulates here above the liquid-air interface refers to, or we can refer to that as let's actually use the first term, vapor density, which is basically the concentration of water molecules in the vapor form in a particular area. And that's more or less related to a more relevant term called vapor pressure. And vapor pressure equals the pressure of vapor on surrounding cellular surfaces, on the surrounding cell walls, for example. All right, so when we have evaporation, when evaporation equals condensation, or the rates of these two, when those are equal, then we would say that that equilibrium has occurred, and equilibrium is referred to as, or let's say the vapor pressure that is present at equilibrium is called the saturation vapor pressure. So we're going to erase this word and call it at the saturation vapor pressure. Okay, now what happens if we increase the temperature? Then we have an increase in evaporation because there is more liquid molecules, liquid water molecules, that have enough energy to enter the vapor state. All right, so that's going to lead to a new saturation vapor pressure. So a change in equilibrium. So when the temperature rises, then equilibrium could be, no longer the state of things, because now there's enough water molecules to continue to evaporate, which is going to increase the saturation. All right, so now that we have water in the vapor state, then the second stage would be, or second driving force is, for evapotranspiration is diffusion. All right, so what is the driving force for diffusion of water vapor? That's our next question. And the answer is called, it's basically like what drives the rate of diffusion, which was a concentration gradient or gradient in chemical potentials. In this case, it's a vapor pressure gradient. So where the vapor pressure is, and we can abbreviate that like this, vapor pressure gradient, where the vapor pressure is high at one area and low at another area. And so where does that vapor pressure gradient occur? From the substomatal space inside the leaves, where the vapor pressure is high, to the atmosphere, outside of the leaf where the vapor pressure is low. In the substomatal space, we can leaves typically have a relative humidity of 100%, which reflects the vapor pressure. Okay, so out in the atmosphere, typically the relative humidity is less than 100%. Not that it never reaches 100%, but if the relative humidity in the atmosphere is 100%, then there is little to no transpiration happening. So this next figure shows us a cross-section of the leaf where we've got the upper epidermis, the lower epidermis. We have the palisade versus the mesophyll layer. You can see the vein and the xylem with water diffusing out to the mesophyll apoplast, and that's that surface that air liquid surface here that is leading to evaporation of water. So the vapor pressure is going to be highest here in the substomatal space, and again lower out here in the atmosphere. Okay, so we talked about all the good things that transpiration does for the plant in terms of circulating nutrients and water, providing turgor for structure and growth. But remember that transpiration can lead to plant water stress, because if it just remains unchecked, then the water supply in the soil is going to run out. So the plant has to have certain things that keep transpiration rates in check. So what are the factors that limit transpiration rates? The first one that we can consider is the stomatal opening or aperture. Okay, so stomata can be completely closed, they can be partially closed, and they can be open, or they can be completely open. Okay, and the more closed the stomata are, the smaller the aperture, in other words, which is the opening, then the higher the stomatal resistance. So the resistance prevents or limits the amount of water vapor that can actually exit from the substomatal space out into the atmosphere. And the triggers for stomatal closure are things like higher CO2 concentration in the atmosphere, a high vapor pressure gradient to limit that water loss from the leaves, certain hormones that might trigger stomatal closure for drought or water stress. We'll write that, hormones. And so that's how the plant is going to respond to, these physiologically to water stress. There is another factor that limits water loss, which the plant can indirectly control, but more or less is a feature of the leaf, and as well as climate, and that's the boundary layer thickness. So in this case, let's say thicker, the thicker the boundary layer, then the greater the boundary layer resistance. Now, what is the boundary layer? The boundary layer you can see here is a layer of dead air, meaning non-moving air across the surface of the leaf. So air tends to push water molecules or water vapor away from the surface of the leaf, but right in contact with the leaf surface there can be a layer of high vapor pressure, essentially, that doesn't move away from the leaf. And so it limits that water vapor pressure gradient. So a higher boundary layer resistance, or a thicker boundary layer, is going to lead to a lower transpiration rate. Just as stomatal resistance, a high degree of stomatal resistance leads to lower transpiration rates. So this leads to the calculation of transpiration rates, the various things that we've discussed here, which is determined as the difference in the vapor pressure of the leaf minus the vapor pressure of the air over these resistances, the resistance of the air plus the resistance of the leaf, or you could do those in reverse order. And the resistance are Resistance of the air would be the boundary layer resistance, and the resistance of the leaf would be the stomatal resistance.